Hello, my name is JohannesHugger and I'm a third-year PhD student at the EMBL EBI, and I present today our project Learning a Representation of Cellular Morphology, Self-Supervised Learning of Shape and Texture Characterization of a Cell. And this project is a collaboration with Valentina Zinchenko from Anna Kresuk's lab at EMBL Heidelberg and Detlef Arendt, who is a group leader at EMBL Heidelberg in developmental biology. So in order to motivate the method that we developed, I want to quickly show you the data that we used throughout this project, which is platinum-erased Dumerilli at 6 days post-fertilization stage. And the data set consists of a 3D electron microscopy volume, where you can see a 2D cross-section of it here on the left side. And we also have, in addition to the 3D EM volume, segmentation maps for each of the cells and each of the nuclei of the approximately 11,400 cells. So the goal here was to find a versatile representation of the cellular morphology. And in this project, when we talk about cellular morphology, we mean the 3D texture of a cell, so what's inside, and the 3D shape that we have through the segmentation mask. And what we want to do, we want to, based on the texture and the shape, compute a feature vector of the cell that is reflective of the cell type. And when we process all the cells of the animal, we want to create essentially a feature space in which cells of similar morphology are close to each other, and cells of dissimilar morphology are further apart. So the approach that we took is based on deep learning, because neural networks are very powerful in visual representation learning. And more specifically, one of the key ingredients that we used to train our neural networks was contrastive learning. And I will explain here how we used contrastive learning on the example of cell shapes. So the way we used it in this pipeline was we sampled a cell, we extracted its cell shape, and then we computed, based on this anchor sample, 2 different geometric views. So we augmented essentially the surface, deformed the surface in a way that it looks different from the original sample, but at the same time, we didn't want to destroy the geometric cellular identity, essentially. So what we did then, we processed both of these 2 samples, augmented samples, with a neural network, computed feature vectors of them, and sampled a random cell of the animal as a negative sample, so to say, extracted the shape, processed the shape with the with the same neural network, computed the feature vector, and we got 3 feature vectors essentially. And what we did then is we had a loss function that essentially encourages the network to embed the two feature vectors that are computed from the 2 augmented positive views close together in feature space, so minimize essentially distance between them, and maximizes the distance between these 2 feature vectors and the feature vector of the negative sample. So this is one of the main ways to train the network. But how does the complete pipeline look like, especially at inference stage? So, for example, when we want to process all of the cell of the animal, we essentially sample each cell, extract its shape, its texture context, which is a large chunk of the cell essentially, but in a more downsampled coarse resolution representation essentially. And we sampled from inside the cell high resolution, so fine texture patches. And all of these 3 things we processed with different neural networks, to compute feature vectors that we then concatenated in one large feature vector. And this is then our final morpho feature representation. Now, so this is our pipeline, but how do we know that we actually learned the right thing, that our features have these properties of reflecting cell type and that similar, morphologically similar cells are close to each other in feature space? In order to evaluate this and make sure that this is the case, we designed different experiments. So the first one was a classification task. And here we labeled approximately 400 cells in the animal. We grouped them into 7 different classes. We have ciliated cells in there, which the animal used to swim, with neurons, muscle cells, secretory, neurosecretory, midgut, and epithelial cells. And we then computed for each of them, for each of these cells, its morpho feature representation, and trained a logistic regression model with cross-validation on it. And then we assessed the accuracy, which is 96% and actually pretty good. Another way to actually qualitatively assess this was then to use the trained classifier to predict the probability of each cell. So how likely it is that a cell belongs to a certain cell class that we decided to group them in. So for example, here on the left side, you can see the probability of each cell in this 2D cross-section belonging to the class of epithelial cells. You can clearly see that on the outside of the animal, the likelihoods are very high because they are mostly located here in these regions. And what was very encouraging and good to see for us is that the classifier assigned a very high probability to these cells here in the foregut opening, which were not part of the test set, but after checking them, they're clearly epithelial cells as well. So we can also do this for the neurons, for example. And yeah, you can clearly see here, here we have neurons which are located in the head, in the ganglia of the animal. And what was cool to see for us, our classifier was able to identify enteric neurons here in the midgut, which were also not part of the test set. So this is all. This was all very reassuring. Another thing that we wanted to check is, of course, if the embedding space or the feature space that we created actually, in a way, groups together self-similar morphology. In order to check that, we essentially computed the morpho feature representation of each cell in the animal, and then plotted a UMAP of it, which you can see here. The labeled dots, the colored dots corresponds to the 7 cell classes that we labeled in the test set. So blue, for example, are neurons, and red are the muscles. And what we then checked essentially is we sampled a cell in this morpho space and check its 3 closest neighbors. So for example, if you take a look at the 4 cells in the upper left part of the figure, you see here the cell that we originally sampled, and those are the 3 closest neighbors in feature space, and they're indeed morphologically very similar in terms of shape and texture. And when you compare them, for example, with a cell belonging to a different class, for example, muscles, clearly different and they are far apart. But what was also very reassuring for us to see is that when we check a cell belonging to the same class of neurons, for example, here, which are here in this figure directly next to them, they are indeed morphologically different in terms of shape and also in terms of texture, actually. So another thing that we wanted to check is if we can actually interpret some of the features that we learned. And here I took an example from the shape features, and I selected one specific shape feature and visualized the cells that have a minimal value of this certain feature and the maximum value of this certain feature. So these 4 cells on the left, which are neuron somata, corresponds to the minimal value of this feature, which can be characterized as essentially these small, compact, roundish cells, and the maxima corresponds to these long, flat muscle cells, essentially. So we clearly can also interpret some of the features that we actually learned. Another part of the project was to see if we can incorporate cellular neighborhood information into the features. And we had different approaches to do this, but I think the one that is a nice combination of simplicity and, let's say, good properties was an averaging approach. So what we did is essentially we sampled a cell and its neighboring cells, computed the morpho feature representations for each of them, and then averaged all of these feature vector into one average feature vector, which essentially then characterizes the cell plus the neighborhood. And we concatenated then this average, let's say, morpho context vector with the original morpho feature vector of the cell. And when we do this for all the cells and visualize them as a UMAP, again, we essentially get clusters not of a cellular morphology similarity, but of, let's say, tissue morphology similarity. So for example, this cluster here in the UMAP corresponds to the cells in the foregut here. And the same here for this cluster on the right. In the UMAP, this corresponds to these clusters of neurons. So in summary, our representation of cellular morphology incorporates both shape and ultrastructure characteristics, captures morphological peculiarities on a finer scale, corresponds to visually understandable features, and when combined with neighborhood information, reflects the morphological similarity between tissue regions. Now, I want to thank Valentina for working with me on this project, and of course our advisors, Virginie Ullmann, Anna Kreszuk, and Detlef Arendt, and of course my group, the Ullmann Lab, and Anna Kreszuk's group. And thank you all for watching.